Hi, class. How is everybody doing? So we are entering into the next chapter, which is one of the most important topic of biology, and that is control of gene expression. So before I go into this chapter, I would just like to explain what does gene expression means. Okay. So gene expression. So we all know that central dogma of biology is that DNA transcribes into RNA, serving as a template, and then RNA translating into protein. That we have studied in the last few chapters, uh, particularly the last one. So what happens that when DNA? So remember that DNA is the body, executive body, from which information is relayed in the form of RNA, right? So, I mean that information or the secrets or the plan of our body is embedded in the DNA. And DNA cannot go every part of our body, right? So it stays in the nucleus, but it has to send out messages. And one of the mode, or the only mode, is that DNA transcribes into RNA and RNA translates into protein. Now, gene expression would mean that whenever a DNA, right, converts or serves as a template to transcribe RNA or translate protein, these RNA and protein both are the end products of DNA. Both serves as a message. Now, you can ask me that why you say RNA? Because I say RNA because not all RNA is translated. Not all of them are, or not all of the RNA is translated. For example, introns, they're not translated, right? But introns carry a lot of messages that controls the protein expression. So, transcription is one form by which DNA and a segment of DNA that has the capacity to express RNA or RNA further leading to protein is called as gene expression, right? So RNA is an expression of DNA. Similarly, protein is also expression of DNA, but in case of protein, RNA serves as an intermediate, right, molecule. mRNA, rRNA, tRNA, ribosome, they all work together as a team to synthesize protein. So gene expression essentially means protein synthesis, and but the exception is that even RNA is a part of gene expression, right? Messenger RNA or rRNA, because there is these RNA molecules sends out some message to a specific cell, to a specific program that is going on in our body. So gene expression, remember, it includes transcription as well as translation. Not only translation, transcription also. And it would include tRNA or mRNA and introns, microRNA, everything. Okay, so that's gene expression. Now, the second term that is being used in this chapter is control of gene expression. So why it says control of gene expression? Why do we need to control, right? So let me give you a very simple example. You all are familiar with New York City. So it's a big city. It's a very busy city. People are moving above the ground, below the ground, and below the ground through subways, above the ground through various means, car, cycle, bicycle, uh, buses, many things, right? So just imagine that if there are no traffic lights on the streets of New York, what's going to happen, right? So for the safety, for the smooth running of New York traffic, or not only New York, all over the world, now we have traffic lights, right? So what do you deduce, what do you see in traffic light? In traffic light, we know that if it is red, we have to stop, right? So what will happen if somebody doesn't stop? Then it might crash, right? The car might crash. It could have a bad consequence. So, and green light would mean that go. Means the traffic can flow smoothly, right? And then yellow light would mean either prepare to start or prepare to stop. Mainly prepare to stop, yellow light, right? It's an indication that soon it will be the red light. So control your speed, right? So what word did I use? Speed. In a car, how do you control the speed, right? First you accelerate, right? Then second, you put brake, right? But before you do either accelerate or brake, you have to start the engine. So start the engine, right? To turn the key around. So first we have to start the car, then push the gas to make the car moving. And if there is any signal, red signal, that either you have to slow down or you have to stop. So we control our car or vehicle through either acceleration or through brake, right? So why I'm saying this, that in our body also there is traffic, right? That is flowing 24/7, just like New York City. And so it has to be controlled. If it is not controlled, the traffic within ourselves, then there will be huge wastage of resources. And if the resources are wasted, then it can lead to disease or certain abnormality, right? So the control of this gene expression occurs at these stages, right? These stages. These three stages, either DNA can be stopped, that don't make RNA. We don't need the protein. Or if RNA is there, okay, the protein is sufficient, the RNA should not undergo translation, right? And vice versa, or reverse. If there is no protein, then the DNA has to push the gas, excuse me, and the mRNA and protein has to be expressed at a very high level or at a high speed. Why? To suffice the need of our body, right? So this is a very dynamic, flexible process that works on the need of our body. And when I say body, it means physiology, right? So that's so let's go back to the slides. So in the book, this is chapter number 8, and it is from pages 261 to 286. On the syllabus, it says as chapter 6. So in the book, it is chapter number 8, and in the syllabus, it is chapter number 6. Okay, and I mean we are coming close to final exam. I'm sure you have a lot of questions about quiz, tests, and future before the exam. So, please take advantage of my office hours, which are also listed on the syllabus, right? So I have on Tuesdays, I have on Thursdays, and also on Wednesday. So I have my office hours 3 days a week. So please take advantage of my office hours, very important. And I get a lot of feedback from students. It's that, I can understand what's going on, what are you thinking in this virtual world, which is very important. But okay, so what's the objective of this chapter? To understand the importance of transcription and translation, right? It's not one-dimensional that DNA will transcribe and protein will be translated, but there are many multi-tier process that affects this simple traffic from DNA to RNA to protein. There, our body is very sensitive. Our body has to be very responsible as we grow right from the day of zygote. So transcription and translation plays very important role, but just being important is not enough. It has to be regulated. It has to do the right things at the right time, right? It's just like our body is just like orchestra. The things have to proceed in a very timely fashion. So we will study the mechanism, what regulates transcription and translation. So let's go ahead. So we all know that our journey starts as a single cell zygote, single cell, then it becomes a multicellular uh, body, and then it becomes spherical. And then, I'm not teaching embryology, but at certain stage, 3 layers are formed: ectoderm, endoderm, and mesoderm, right? And after these 3 layers are formed, this is very important, that these 3 layers are responsible for formation of various kinds of cells. Cells form tissues, and tissues, they lead to the formation of organ and organ system. For example, let's say endoderm, it forms lung cells, thyroid cells, digestive tract, or pancreatic cells. Now, the point I am mentioning to you here is that we start our journey as a zygote, right? And the DNA in the zygote is exactly the same at the 8-cell stage embryo, then at blastula, then when the 3 layers are formed. Then if the DNA is same in this journey of embryo, but the interesting part is that how come the zygote, the cells that carry the same DNA, how they develop into lung cells, thyroid cells, right? Digestive cells, cardiac muscles, kidney cells, red blood cells. Now, just let me ask you a question. Just because all the cells emerge from zygote, can one of them replace each other? No. What do I mean? That let's say if there is a failure of kidney, can RBC replace kidney? Ideally it should, right? Because they carry the same DNA. Or can muscle replace kidney? It's difficult, right? Because despite they carry DNA, they are functionally very different. That begs the question that if the cells is originating from one point, zygote, how they become skin, how they become neuronal cells. What are the functions of neuronal cells? Memory, to feel pain, to communicate, senses. So can thyroid cells replace neuronal cells, or vice versa? Can they replace each other? No, it's very hard because they're very specialized. So what are the mechanisms that helps the cells to become more specialized? So that's the basis for gene expression, and that becomes and the gene expression has to be controlled, just like New York traffic light, because if it is not if the traffic is not regulated, either people will go late, reach late, or there will be chaos or maybe accidents, right? So the traffic in our body also is very much tightly regulated, such that all the deadlines are met. What are the deadlines? So the deadlines is that in a certain day, the zygote has to become 8-stage, then into blastula, then blastocyst, and 3 layers. If these deadlines are not met, then there can be developmental delay, and they can lead to some kind of disorder. So the next slide is, I advise you to watch this YouTube video. It's very interesting, and you will get the idea of this whole chapter. So one thing is, I gave you an example of traffic light and how embryogenesis takes place and give rise to a specialized body of human body, starting from brain to skin to nails, right? another very important question that I want you to think, and these are facts, right? So look at the size of a human body and flies. There's a huge difference, right? So what would you expect? That the genes that makes protein in human body should be million times more than the flies, right? If you compare the body, anybody can imagine that human body is huge. There are many functions. There are many kinds of cells. So the difference between these two should be huge. But the data reports that humans have approximately only 24,000 genes, but fruit flies have only 14,000, just 10,000 difference, which is very small. So why? What's going on? So scientists found out that human proteins do multiple functions. So I repeat again, the difference of the genes between fly and human being is not much, is because human proteins, they do multitasking, they do multiple functions, right? Second thought, the third thought I would like to also you to know is that let's say our DNA, let's say our DNA is 100%, right? So that is present in 46 chromosome or 23 pair of chromosome, the DNA that is present, only 2 to 3% of the DNA makes protein, right? Only 2 to 3% of the DNA make protein. So what do 97% of the protein DNA does? So scientists thought that these 97% of the DNA is a junk DNA. It has no function. But with the development of technology, scientists realized that this junk, this 97% of DNA is not junk, but it does regulatory functions. And what are some of the regulatory functions? One of the most important regulatory function is control of gene expression, right? So what examples I gave you before we start this chapter, go deep into this chapter. Example of New York traffic, right? Red light, green light. You know, some of the pathways or some of the streets are one-way street. People can only move in one direction, the vehicle, the cars. Some streets are two-way, right? In some streets, there are avenues and street, just like Eastern Parkway when we come to our school, right? So it has to be regulated, right? The other example I gave you was that our development. Why, how do cells acquire different functions despite they carry identical genome, identical DNA? Third or fourth example I gave you about the difference between the size of fly and a human body. But the number of genes are not significantly different, only 10,000, which suggests that protein has many functions in humans. And one of those functions could be control of gene expressions, right? Then we talked about junk DNA, that 97% of the DNA in our cells, they may not code for protein. So what's their job? Their job is to control the gene expression. And what is gene expression? The gene expression is when DNA serves as a template to either transcribe and translate both. That DNA activity is called as gene expression. Okay. So this slide shows what are the steps at which genes could be regulated. So focus right on this slide. This slide is the I have used this slide many points in the course of this lecture, but pay attention, right? The starting point is DNA, right? DNA serves as a template for transcription and then transcription, then translation. So this step between DNA and RNA, this is called as transcription, right? But this step between DNA and RNA, this transient step is called as, can be controlled. So this can be called as transcriptional control, of gene expression. After the fresh RNA is formed, this RNA undergoes modifications like capping, poly-A tail, splicing. So this transition from primary transcript to mature mRNA, this is called as post-transcriptional regulation, because this happens after the transcription, but before the mature mRNA is formed. And when the mature mRNA is formed, it is ready to be exported to the cytoplasm to meet the ribosome for translation. Now, as I mentioned to you, that if there can be a situation where the mRNA doesn't need to be translated, it needs to be controlled, it needs to be stopped. So this is the fourth stage at which mRNA can be controlled. So transcriptional level control, splicing RNA processing control, export control, and then mRNA can be removed by degradation. Then mRNA can also be controlled for binding to ribosome that we'll study, right? And finally, when the protein is made, that's translational control. It can be modified. What do I mean by modified? That a protein could exist by itself, or it can undergo phosphorylation by a special group of enzyme called as kinase. So this phosphorylated protein can be more active than the unphosphorylated protein. So what is the message here? That phosphorylation can increase the protein activity, protein function. And this we see phosphorylation plays a very important role in cell cycle, right? That we'll study in next chapter. So these are the 6 pivotal points on which the gene expression is controlled. Okay. So let's discuss few terms. For example, let's discuss few terms. For example, repressor, right? The one term I would like to discuss with you is repressor. Repressor is a protein that binds to a portion of promoter. And when repressor binds to the promoter, then it blocks the RNA polymerase to synthesize mRNA, right? So you can think that repressor is just like brake, right? If you put a brake on our car or it will stop. So repressor, and how does it stop? Repressor binds to promoter or a portion of promoter and blocks RNA polymerase. So if RNA polymerase binds to promoter, it will initiate mRNA synthesis or RNA synthesis. But if repressor is already present on the promoter, then RNA polymerase cannot bind and transcription and following translation will not occur. Very contrast to repressor is another protein which is called as activator. Activator, it means like putting the feet on the gas, and it accelerates. It accelerates the production of mRNA and protein, which means that it works as a friend to RNA polymerase. So repressor repressor and activator, they are 180-degree opposite functionally, right? One stops the expression of mRNA and subsequently protein, and activator, it works with RNA polymerase, creates space with RNA polymerase to increase the production of mRNA and protein, right? So repressor and activator. So let's go back to the slide. So repressor turns the genes off. Activators turn them on, right? Like your switch. If you turn it off, there's no light. You turn it on, there is light. So one of the example for the repressor is tryptophan. Now remember, bacteria, they're very smart. They're unicellular, they're single cell. They are extremely sensitive to environment. And they are equally sensitive to the nutrition, right? So let's say if a bacteria is thriving or growing in a nutritional medium where there is enough tryptophan. Tryptophan, what is tryptophan? Tryptophan is an amino acid, right? And one of the functions of tryptophan is that it has been studied that many DNA binding protein needs tryptophan to bind to DNA, right? So it's very essential amino acid, tryptophan. So what bacteria has done to conserve its energy, because bacteria, they're very smart, right? So if there is if there is low tryptophan or no tryptophan around bacteria, right, then bacteria has no choice, right? So bacteria will turn the gene that will make mRNA for proteins that will help in proteins synthesis of tryptophan. I repeat, if tryptophan is lacking in the media or there is low tryptophan and the bacteria needs tryptophan, then it has to switch on gene, right? For the synthesis of, for synthesizing those proteins that will synthesize tryptophan. But if the tryptophan is high in the medium, then bacteria doesn't need to synthesize tryptophan, doesn't need to synthesize those proteins that participate in protein synthesis. So the genes have to turn off. So it is turned off by the repressor, right? So if tryptophan is present, then the repressor binds to the promoter and doesn't let RNA polymerase drive the transcription. In contrast, right? So repressor is driving here. It's a repressor model. If it is present, then no tryptophan synthesis. If it is absent, then mRNA synthesis takes place. So the next slide will a little bit describe to you that if tryptophan is binding to repressor and repressor is binding to operator, but this may be a new term for you. So operator and promoter, they exist together. The repressor binds to the operator, RNA polymerase binds to promoter. If the tryptophan is present, then tryptophan binds to repressor and then repressor binds to operator. When operator is occupied by the repressor, the RNA polymerase cannot move. And if it cannot move, it cannot synthesize proteins or enzymes necessary for synthesizing tryptophan. So it's stopped here. But if tryptophan is absent, then the repressor is off the operator. Then RNA polymerase can slide through the those genes that synthesizes proteins essential for the synthesis of tryptophan. So this is a repressor model. It is driven by the repressor. Now let's go to the next example of activator. So what I mentioned to you on the whiteboard, that activator is accelerator. It has a positive influence on the RNA polymerase. So when activator goes and bind to the promoter, it helps RNA polymerase to synthesize mRNA. So activator and promoter, they are like opposite sides of same coin, head and tail. So when activator binds, RNA polymerase do the job. Now let's look at the example, right? So one of the example is the lactose model, right? So bacteria, they like lactose, right? So what happens that if lactose is present in the medium, right, then the bacteria needs to utilize the lactose through three genes: beta-galactosidase, permease, and trans-acetylase. I repeat, when the lactose is present, it needs to metabolize lactose through these three genes, right? Three proteins: galactosidase, permease, and trans-acetylase. But what happens that it needs an activator, right? So the allolactose acts as an activator that removes the repressor from the operator, and then the RNA polymerase synthesizes these three genes. So when lactose is present, it serves as a activator, inducer. So let me just recall, if tryptophan is present, then tryptophan acts as a repressor. When lactose is present, it serves as a it has a positive influence, right? So let's look at this slide. So pay attention here that when a situation where glucose and lactose, right? Glucose and lactose, they are both present. Then the operon was off. Right? When glucose is present, this promoter region is occupied by the repressor, and the operon is off. When glucose and lactose both are absent, right? Then the bacteria is feeling the need to get lactose or glucose. So what happens, this cap protein comes and binds to the region very close where RNA polymerase will bind. And when lactose is present, right, then the RNA polymerase, the cap, it serves as a facilitator for RNA polymerase to bind to the promoter and then start the transcription and translation of three proteins: beta-galactosidase, permease, and trans-acetylase, right? So this is a lactose model, which is inducer-based model. I'll go into what is CAP in the next slide, right? So now remember, don't get confused. When glucose is present, the operon is off. When lactose is present, then the operon works. This is called as lac operon. Okay. Also, please watch in every slide. I have most of the slides. I have a YouTube video. Those are very interesting short YouTube videos that will clarify your concept. So CAP protein, it is an activator, right? So catabolite, CAP stands for catabolite activator proteins, right? It is also called as cyclic AMP receptor protein. Right? Each subunit of CAP is composed of ligand binding domain, and so it has an FCAP binds to DNA upstream of RNA polymerase, and CAP activates transcription through protein-protein interaction with RNA polymerase. So as I mentioned to you, CAP is an activator that works with RNA polymerase to activate the transcription. So now let's review this table. In prokaryotes, in bacteria, there is only one RNA polymerase, but eukaryotes, there are three. The chromatin structure, there are no nucleosomes in bacteria, but human chromosome is tightly packaged into chromatin. And that chromatin, the building block for the chromatin is nucleosome. In bacteria, since it is a unicellular, it doesn't need differentiation. There is, it stays for whole life as a single cell. There is no requirement for general transcription factor. But in eukaryotic cells, we have to not only divide, but we have to develop into specialized cells. So it requires transcription factors. Transcription factors is very helpful for cell fate determination, right? I mentioned to you that when zygote is developing, it differentiates into specialized cells. So transcription factors help these the RNA polymerase for the cells to differentiate into specialized cells. And regulatory regions, bacteria has only one promoter, whereas eukaryotic chromosomes, they are very complex. So they have promoter, they have enhancer, and they have also long distance communication. So I want to clarify here what is enhancer. So enhancer is a region that is upstream of promoter, right? So what is promoter? Promoter is a sequence of DNA to which DNA polymerase bind. It is upstream of the start site, right? Promoter is upstream of the start site, and promoter is made up of at -10 theta box and -35. Now way up almost 1,000 to 2,000 base pairs upstream of promoter, there are specialized sequences called as enhancer, right? So almost 1,000 to 2,000, in general 2,000 bases upstream of promoter. Now you can ask me a question that it is so far upstream 2,000, how does enhancer communicates with promoter? So if you see here, the DNA, the gene has the ability to loop, right? These looping is mediated by proteins called as mediator. So because of this looping, long distance enhancer becomes very proximal to the promoter, right? And what is, as the name enhancer suggests, enhancer enhances the transcription. An enhancement of transcription would mean increase transcription, increase activity of RNA polymerase. So enhancer comes proximal to the promoter through proteins, mediator. These mediators, they communicate with activator proteins that binds to the enhancer and helps in looping in, right? And then it comes in proximity with the RNA polymerase to initiate the process of transcription. And the example I give you here is of these highways. That highways, they are connected with various loops that connects two different highways, right? And that reduces the time for travel. So if you go through the local traffic, it will be longer because of traffic lights. But if you take highways, it is shorter. And also, if the two highways are connected, then it further facilitates the reduction of time that might take for reaching a destination. Right? So let's focus on chromatin, right? So our chromatin is made up of DNA and protein, right? And what are the proteins? The proteins are histones. How are the histones present? They are present as nucleosomes. And what are nucleosomes? Nucleosomes are 147 base pairs of DNA with octamer of histone proteins, right? So if the nucleosomes are very condensed, right, if they're very tight, then transcription factors, RNA polymerase cannot access the promoter. It is tough to access. So how to relax this chromatin, how to relax this nucleosome so that transcription machinery can access promoter? This is facilitated by histone modifying enzymes, right? So one of the enzymes is that acetylates the nucleosome, acetylates the histone. And when acetylation occurs, as histone acetylation occurs, this nucleosome, which was condensed, becomes relaxed, right? So, and when it is relaxed, this DNA is exposed, which can be accessed by transcriptional machinery. What are transcriptional machinery? RNA polymerase, activator, general transcription factors, mediators. So that accelerates the process of transcription. So histone modifying enzymes and chromatin remodeling complex, they change the landscape of nucleosome to facilitate transcription initiation. Which modification did I talk to you? Acetylation. Where does acetylation occurs? It occurs on histone proteins. And histone, what are proteins made up of? Proteins are made up of amino acid. So which amino acid undergoes acetylation? So it is amino acid lysine that undergoes acetylation. When lysine is acetylated, the nucleosomes, they relax. When they relax, they expose the DNA, the promoter. When the DNA is exposed, promoter is exposed, the RNA polymerase can access the DNA. And that leads to the increase in transcription. Right? So until now, we have talked about tryptophan repressor system. We talk about LAC or lactose-based inducer system. Then we talked about enhancer. How does enhancer come closer to the promoter through mediators and transcriptional activators that enhances transcription. Then we talked about lysine acetylation and chromatin remodeling that changes the nucleosomal, that relaxes the nucleosomes and helps the transcription. Now let's focus on this slide. So pay attention here. So if DNA is methylated at cytosine, right? If DNA is methylated at cytosine, then there won't be any transcription. So let's say if here both the strands are methylated at nucleotide cytosine. Remember that cytosine undergoes methylation. So when DNA replicates, it gives rise to daughter cells, right? So one of the strands is still methylated, right? So this methylation of cytosine, right? It blocks the process of transcription, right? So a methylated DNA will not transcribe because methylation stops the access of DNA or promoter to the RNA polymerase and transcription machinery. So just like a red sign of traffic light that says stop immediately, methylation is also a stop sign for the DNA to not to transcribe. Right? So let's again revise. We talked about DNA by DNA methylation. This is how transcription is controlled. If DNA is methylated, then no transcription, right? Let's say a primary transcript is formed, right? But this can be regulated by tryptophan and lactose, right? Lactose is an inducer, tryptophan is a repressor, right? Also, if DNA of nucleosome is acetylated, then the transcript will be formed. So these are, so DNA methylation, acetylation, enhancer proximity to the, it controls the primary transcription. So those are transcription control. So let's enhancer, inducer, repressor, acetylation, methylation of DNA, they are part of transcription control. Now let's go into post-transcriptional control. Post-transcriptional control, mainly we will talk about splicing of RNA. So, I'm hoping that you're focusing. So see here, this is a gene, right? This gene has exon 1, then intron, then exon 2, 3, 4, 5, and 6. So there are introns and there are 6 exons, right? So let's say this is the primary transcript. Primary transcript means it has to undergo splicing to become mature mRNA. So if the splicing occurs, that leads to the joining of exon 1, 2, 3, and 4, right? Then if the splicing occurs in this fashion, where exon 1, 2, 3, 4 comes together, this forms a protein called as calcitonin, right? So this calcitonin is mainly required in the thyroid function, right? And another scenario happens where 1, 2, 3, 5, and 6 exons, they come together to form mature RNA. This forms a calcitonin gene-related peptide. So differential splicing of a single gene, right? This is a single gene where there are 6 exons, right? So these exons can be spliced and ligated in 2 different fashions. In fashion 1, 2, 3, 4, it gives rise to calcitonin. In order 1, 2, 3, 5, 6, it gives rise to CGRP, calcitonin gene-related protein. So same gene can undergo different kinds of splicing to give rise to 2 different kinds of protein which have 2 different functions. So this is a post-transcriptional control. So splicing is a post-transcriptional control. Now, this could be new for you, but this is now becoming very important topic already, called as microRNA, right? So what are microRNAs? This is a group of small non-coding RNAs. What does non-coding means? That these RNAs don't express protein, but they can control gene expression. And it was discovered in plants as well as animals. In humans, there are about 400 microRNAs, right? And these microRNA, they are very short, 20 to 22 nucleotides long, and they can bind to RNA. And when these microRNA can complementarize with mRNA, and when they complementarize, the mRNA can be degraded. So if an mRNA can be degraded, then there will be no protein synthesis. Now this miRNA, it exists in our cells as a precursor molecule. Look at the structure. They're like hairpin structures. They are loops, and they are at some places loops because there is no complementarity, and at places where they complementarize the 2 strands, they become double-stranded. So after it is synthesized, so this is an RNA, so this is synthesized inside the nucleus, and then it is exported to the cytoplasm. In cytoplasm, there are some proteins which are called as RISC protein, RNA-induced silencing complex. RISC stands for RNA-induced silencing complex, right? So these RISC proteins, they convert this double-stranded RNA into single-stranded RNA. And this RNA, which is miRNA, almost 22 nucleotide in length, they complementarize with designated mRNA. And then this mRNA is spliced off. When mRNA becomes double-stranded, it can be targeted by enzymes that can chop this mRNA into pieces such that there is no protein synthesis or translation, right? Now this has huge therapeutic significance because there are many genetic diseases where a different or a wrong protein is formed. For example, in sickle cell anemia. So this is a genetic disease. So in sickle cell anemia, the gene is defective, there's a mutation, and the hemoglobin is defective. So how to treat this? Or there's also some genes such as muscular dystrophy, where a gene is affected and it affects the protein functions. So on one hand, defective mRNA can be targeted. On the other hand, normal proteins could be used as a supplementary in these patients to treat diseases. So miRNA strategy is becoming a very important therapeutic strategy that you'll hear soon, especially in cystic fibrosis also. So miRNA, so read about miRNA risk complex. It targets the mRNA that prevents translation. So this becomes, this is happening in cytoplasm, right? So again, I'm again emphasizing that watch this video on miRNA. So when mRNA is targeted such that there is no expression of protein, there is no protein synthesis, this process is called as RNA interference, right? And what is the protein involved? RISC, RNA silencing complex. Right? RNA, in the last slide, RISC is called as RNA-induced silencing complex. So let's see what did we discuss until now. We discussed about DNA, where we talked about tryptophan, lactose, methylation, enhancer, and acetylation. Right? Then we talked about RNA processing control, where one gene which has 6 exons can be processed in 2 ways, giving rise to 2 different proteins with functions. Then we talked about mRNA degradation control, right? Through miRNA. So it can make the RNA inactive. Now let's talk about translational control, how to control the translation. So this is very interesting. So focus here. If AUG, so what is AUG? AUG is the start codon. If AUG is linear, then ribosome can bind, right, and start translation. But if there is a protein that is occupying the ribosome binding site called as translational repressor protein, then there is no translation, right? So ribosome cannot remove this translation repressor protein. So this is a sort of a translational control, one way. The second way is that, let's say the AUG is not linear, but it is part of the hairpin. So you see, this is part of the stem of the hairpin. So ribosome cannot access this AUG. So there cannot be translation. But if this AUG is, if AUG becomes linear and the ribosome can access this AUG, then the protein is made. Same concept here. If AUG is linear and available to ribosome, protein is made. If it is part of that it is not accessible to ribosome, then there will be no protein made. So AUG has to be linear. AUG and ribosome binding site has to be linear so that ribosomes bind and then translation occurs. So remember how translation occurs? Ribosome binds and it slides across the mRNA and protein is made, right? Sometimes there is also another strategy called as antisense RNA. If antisense RNA is complementary to the mRNA and mRNA becomes double-stranded, then again, this is a RNA interference and then no protein will be made. Yeah. So again, we talked about, just to review, we talked about transcriptional control, we talked about splicing for RNA processing control, we talked about translational control, ribosomes can bind when ribosome binding site and AUG is is linear. If it is not linear, there is no translation. The last is protein activity, right? So let me give you an example here. So let's see that, go back to where we started. DNA can transcribe into RNA, then RNA into. So what happens now that this protein can exist in two forms, right? This protein can exist in two forms. One is unphosphorylated, and the other form is phosphorylated. Now, how does this happen? This happens because of an enzyme, right? By enzyme, let's say for sake of more simplicity. So this can exist in a dynamic form, right? This can exist in a reversible form. So an unphosphorylated protein can be phosphorylated with the help of enzyme called as kinases, right? And a phosphorylated protein can become unphosphorylated by the help of enzyme called as phosphatases. So kinases, they phosphorylate, and phosphatases, they remove the phosphate group, right? So they remove the phosphate group. And what is the phosphate group? Is PO4, right? So it has been seen that there are some proteins which, if phosphorylated, they become, if they are phosphorylated, I repeat, they become active. And if they are unphosphorylated, they become inactive. You know? So phosphorylation is governing. So the same protein, but it can exist in two forms, unphosphorylated and phosphorylated. So phosphorylated, it increases the activity. So phosphorylation has a capacity to change the functioning of a protein, right? This is the basis of signal transduction that we'll study in cell cycle in cancer in the next chapter. So going back to the slides. So there are some, as I mentioned to you, that there are some proteins. So for example, what happens in signal transduction? In signal transduction, cells send some signal outside the cells on them, and this signal outside the cells is transduced inside through receptor, right? But it from outside to inside, post binding, in this case insulin binding to insulin receptor, it brings changes in the receptor, and this changes is communicated through the membrane, and then there's a cascade of events that takes place and goes to the nucleus. So insulin binding to its receptor is communicated to the nucleus through a cascade of communication between proteins. And one of the communication is through phosphorylation and dephosphorylation, right? That we will see in the next slide. So insulin binds to its receptor, and see there is a range of phosphorylation that occurs. So this is an insulin receptor. It has extracellular domain, it has intracellular domain. The moment insulin binds, the intracellular domain is phosphorylated. This phosphorylated proteins serves as a switch, and it has a ripple effect, right? And then it activates the downstream proteins through phosphorylation and dephosphorylation. Ultimately, this what is happening in the extracellular environment reaches to the nucleus. And in the nucleus, a subset of genes are transcribed and translated. And this process, where an extracellular signal is passed on through cascade of proteins through phosphorylation or dephosphorylation, and then the cellular genome or DNA responds to this extracellular changes. This is called as signal transduction, and this is mainly through phosphorylation and dephosphorylation. So please again watch these videos on signal transduction, and these are some of the quiz questions that you can review. So with this, we finish this chapter. Thank you.